Hello, my name is Lauryn Magwaro, and I'm currently a junior at Hamline University majoring in biochemistry with a minor in digital media arts. This summer, I was able to partake in Hamline's summer collaborative undergraduate research program, and I was able to collaborate with Dr. Betsy Martinez-Vaz, along with her collaborator, Dr. Lawrence P. Wackett at the University of Minnesota. I was also able to work with peer collaborators, Dean Young and Anna Schultz. Our title was Substrate Specificity of Guanylyurea Hydrolase Across Multiple Microbial Species. Before getting into my research, I will give a small background. Metformin is one of the most prescribed medications in the world, with over 80 million prescriptions per year in the US alone, and its use is expected to increase as the global prevalence of diabetes rises. Once taken, metformin is not metabolized in the body and finds its way into wastewater treatment plants. Guanylyurea, metformin's transformation product, is said to be a dead-end metabolite that is accumulating in water. However, recent studies found that there is an enzyme, guanylyurea hydrolase, in Pseudomonas medicina strain that is able to break down guanylyurea into guanidine and ammonia. This enzyme catalyzes the second reaction in the metformin biodegradation pathway, which can be seen under the background in Figure 1. To further investigate the use of this enzyme, research was conducted to investigate 13 potential bacteria that have similar enzymes to that of guanylyurea hydrolase. A blast search identified proteins homologous to guanylyurea hydrolase enzyme in 13 different microbial species. The microbial species that we were working on are in the middle center of the poster. Plasmids containing the genes encoding the guanylyurea hydrolase homologs were transformed into E. coli. These cells were then induced to simulate protein production and prepare cell extracts. We were able to check for induction using an SDS page analysis, checking for induction. You can see that in the upper right corner of the poster, we circled where the bands were thick, showing the 25 kilodalton induction of guanylyurea hydrolase. After that, protein concentrations were determined using the BCA assay, while enzyme activity was tested by measuring the amount of ammonia release using the Berthelot reaction. So the BCA was the purple that can be seen under methods, and then the Berthelot test was the blue enzyme test that can also be seen under the methods. The substrate specificity of the guanylyurea hydrolase homologs was investigated on guanylyurea, as well as similar compounds, including bioret cyanoguanide and 2-amino-4-thiobioret. These 4 compounds can be seen towards the upper right corner of the poster under Figure 8. Out of all the 13 bacteria strains tested, only 8 of them had activity with guanylyurea and or bioret, and no enzyme activity was observed with cyanoguanide or 2-amino-4-thiobioret. This can be seen under the graph in Figure 9. That said, most guanylyurea hydrolase homologs can only degrade guanylyurea, implying that guanylyurea hydrolase has a narrow substrate specificity. Future steps will take a look at purifying for further optimization of yield and purity, but also determining optimal conditions of catalysis. That said, also additional substrates could also be tested in order to determine enzyme substrate specificity. Thus, the goal of this study was to investigate the level of enzymatic activity and substrate specificity of the guanylyurea hydrolase homologs identified across the different microbial species. That said, it was an amazing experience to be able to work on this project this summer, and I was able to learn what it means to be a scientist and also how to analyze and collect data. With that said, thank you so much for listening to this project, and I hope that you learned something interesting about bacteria.